Hi and Assalamualaikum. My name is Nurul Ain binti Muhammad Liki. My number matric 23 DBT 20F 1012. I'm from DBT 3A. Today, I will do lab simulation for enumeration of bacteria, which is viable plant count. The viable plant count, also called a standard plant count, is one of the most common methods for enumeration of bacteria. This is the introduction for viable plant count. First of all, we should take seed dilution tube. Each contains 9 ml of sterile saline. Dilute 1 ml of sample by withdrawing 1 ml of the sample and dispensing this 1 ml into the first dilution tube. Using the same procedure, withdraw 1 ml from the first dilution tube and dispense into the second dilution tube. Subsequently, withdraw 1 ml from the second dilution tube and dispense into the third dilution tube. Continue doing this from tube to tube until the dilution is complete. Next, transfer 1 ml from each of only the last 3 dilution tubes onto the surface of corresponding agar plant. Incubate the agar plant at 37 derajat Celsius for 48 hours. Choose a plate that appears to have between 13 and 300 colonies. Count the exact numbers of colonies on the plate. Calculate the number CFUS per milliliter of original samples as follows. First of all, click on dilution tube wrap of test tube to bring them to the table. Each of the dilution tube contains 9 ml of sterile saline solution. Next, we should click on the water sample to bring them to the table. Now click on the eye drop to withdraw 1 ml of sample, 1 contain water, and dispense this to the first dilution tube. Click next to ensure sample is finished. Next, the dilution for each of the 6 dilution tubes can be summarized in the image below. Dilution tube 1 has 1% dilution with dilution factor of 10. The dilution factor for each of tube is listed below. Tube 1, this. Next, we will be enucleating agar plate with the last 3 brood culture dilution. Click on the agar plates on the shelf to bring them to the table. Now, click on the ice drop to transfer 1 ml of dilution 4 to plate 1, 1 ml of dilution 5 to plate 2, and 1 ml of dilution 6 to plate 3. Next, click on the pencil to label agar plate 1 with dilution factor 10000, plate 2 with a dilution factor 100000, and plate 3 with a dilution factor 100000. After that, click next and click on the agar plate to place them in the incubator at 37 derajat Celsius to 48 hours. We will now need to perform the same dilution and enucleation step for each of the test sample. The process is the same for each sample, and we will assume the process of dilution and inoculation has been complete for all 4 of the water sample, and the 48 hours of incubation times has now been complete. Click on the incubator to bring all of the insulated agar plate to the table. Each of the group of insulated plate is labeled with the source of their respective sample. A key for the sample is given below. This is the result for fountain water. Is a colony from this we can see have 36 number of colony. For this have 7 number of colony, and for this have only 1 number of colony. Second, for result for boiled fountain water, have 45 numbers of colony. This one have 2 number of colony, and lastly don't have numbers of colony. This for result river water. From this we can see have 42 numbers of colony, and this have 13 numbers of colony. Next, for boiled river water, these have 45, 54 numbers of colony. These have 8 numbers of colony, and lastly this one have 3 numbers of colony. That's all.